We're going to talk about complex genetics, incomplete dominance, codominance, and multiple alleles. Here's your learning goal for today. Construct a Punnett square to determine the genotype and phenotype of a mating involving incomplete dominance, codominance, and multiple alleles. Now, previously we've talked about simple dominance, and that's where one allele is dominant to the other and completely covers up the fact that the recessive allele is there. Okay, and we've used the example before of big P for purple, little p for white in flower color in plants. In simple dominance, the heterozygous genotype shows the dominant phenotype. So in simple dominance, whenever you have a heterozygous genotype like big P, little p, you're going to get a purple flower. And when we do our Punnett square, if we take a homozygous purple flower and a homozygous white flower and we cross them, our Punnett square would look like this, where all the offspring are heterozygous and they're all going to be purple because the purple allele is dominant. That's simple dominance, right? We've done lots and lots of problems like that. Incomplete dominance is different. Incomplete, the dominant allele can't completely cover up the appearance of the recessive allele, so you get a third phenotype when you have a heterozygous genotype. Okay? So in other words, if you have one dominant allele and one recessive allele, instead of getting the dominant phenotype, you'll get a mixture of the dominant and the recessive phenotype. And you'll get a third phenotype, an intermediate, whenever you have the heterozygous genotype. Now sometimes we use this notation to indicate incomplete dominance, where we have two capitals, but one of the capitals has a prime there. You need to be aware of that. It doesn't show up very often, but it's something you should know. So let's look at an example. In incomplete dominance, if we take a red carnation and a white carnation and we cross them, we'll get a pink. We don't get red, we don't get white, we get a third phenotype, right? A pink. So let's look at how that happens. So in carnations, capital R is red, little r is for white. So if we take a homozygous red carnation and a homozygous white carnation and we cross them, when we do our Punnett square, we will get all the offspring will be heterozygous, and instead of being red or white, they'll be a third phenotype. They'll be pink. So the heterozygous form shows a third phenotype. That's your tip-off for incomplete dominance. And another type of complex genetics is what's called codominance. In that, basically, both alleles are dominant. Not only are they both dominant, but if you look, both alleles are capitals. We don't have any lowercase alleles. So we always said that capital letters meant dominant. So in this case, both alleles are dominant. When you see that, when you see both alleles as capitals, that should be a tip-off to you that you're dealing with codominance. If the allele is present, you'll see it in the phenotype. Neither one can cover up the other, and they don't mix together. In incomplete dominance, they kind of mix together. In codominance, they don't mix. Both phenotypes are present. Okay? So the heterozygous form shows both phenotypes. And we're going to look at chickens. If you take a white chicken and a black chicken, you don't get a gray chicken, you get a striped chicken. you get a chicken that has black feathers and white feathers, right? Not gray, but both traits will be expressed in the heterozygous. Okay, so let's look at this. We're going to take a homozygous black and a homozygous white chicken, and we're going to cross them. These are the phenotypes. Excuse me, these are the phenotypes and the genotypes that are possible. You could have a black chicken that's going to be homozygous B, a barred chicken, a stripedy chicken, which will be heterozygous, or a white chicken, which will be homozygous W. So we're going to take a homozygous black and a homozygous white, and we're going to cross them, right? Here's our black chicken at the top and our white chicken at the side. All their offspring are going to be heterozygous. All of them are going to have both black feathers and white feathers. That's the hallmark of codominance. Both traits show up. Okay, one can't cover up the other. Now, multiple alleles, this is something altogether different than what we've talked about. Up to now, we've only had two versions of each gene. We've had two alleles. Now we're going to have multiple alleles. All right? And a great example of this is blood type. We have three alleles for blood type. We have IA, which makes a type of protein called an A protein on red blood cells. Another allele is IB, which makes B-type proteins on red blood cells, and little I, which makes no proteins on the surface of red blood cells. These proteins on the surface of red blood cells make up blood types. Okay, so if you have the A-type protein on your blood cells, then you have type A blood. If you have the B-type protein on your blood cells, then you're type B. And if you have both A and B proteins, then you're type AB. And if you have none of these marker proteins, then you're type O. So this means that for each phenotype, we can have multiple genotypes now. So let's look at the genotypes that go along with the blood types now. So for type A blood, for phenotype A, you could have IA, or you could have IA and little I. Same thing for B. You could be homozygous IB, or you could have IBI. For AB, there's only one combination. You have to have IA and IB. And for O, there's only one possible combination of alleles, and that's two little i's. But for type A and type B, there are two genotypes that will give you the same phenotype, right? These two genotypes will give you this phenotype. So let's look at a problem. Suppose a man with type A blood and he's heterozygous. He has IA, little I, and he marries a woman that has blood type AB. Let's see what blood types their offspring would have. Here's the man's across the top, his possible gametes. He could make a sperm that carries IA or a sperm that carries I. And his wife's eggs down the side, she can make an IA egg or an IB egg. That's it. Let's see what happens. So they could produce these possible offspring. They could have one offspring where the genotype is IA, and the phenotype would be type A blood. They could have an offspring with the genotype IA, little I, right? That would make type A phenotype blood. They could make a type AB, or they could make a type B with IB, little I. Now, one thing you might be interested or curious about is donating blood or receiving blood. If you have type A blood, then you can give blood to somebody with type A or type AB blood, but you can only receive blood from someone who has type A or O. Somebody with type B blood could donate safely to somebody with B or AB, and they could receive from B or O. Type AB can only donate to AB and can receive blood from everybody, from A, B, or O. O blood can donate safely to all, but can only receive blood from people with O blood.